So I first met Harry at a meditation event in April of 2021. Some mutual contact introduced us, said this guy's working on some frequency water, and I'm like, okay, what's water, all right. I was very skeptical, didn't know what was inside this water, how they changed this water, and what kind of frequency information was in it. We met, talked, I was intrigued, and as a scientist, I tend to have a very open mind. And so I said, send me some of that water and we'll take a look at it. It sat on our shelves for a couple weeks because we're busy with a lot of stuff. Eventually we started doing some studies with it, and the findings were unexpected, right? And so as a scientist, when you discover something unexpected, the thoughts are either something went wrong in the experiment and we've got to repeat it, or something went right and we have to repeat it, right? You just have to repeat over and over again. We repeated it multiple times and the same effect came through every time. So the initial experiment we did was a cell biology experiment, right? We put this water that had particular information in it on a cell, and we noticed that the cell behaved completely unexpected from how it should. So then, as a curious scientist, the question for me was, well, what's that information? How is it different? What's inside of it? And could we somehow measure that change in information? So we scoured the lab, found a bunch of different waters, just water right out of the tap, water that everyone drinks in the office area, water from a completely pure distilled source, and then waters from Ness and Harry. And we have a machine in the lab called an electron paramagnetic spin resonance machine. It's a machine that's literally a black box. It's a very fancy microwave. What you do is you bombard a sample inside a little capillary with microwaves, and what happens with this is then if there's any kind of material inside, it can create a spin resonance. And so when we put tap water in, we got this interesting frequency change. When we put normal distilled water, there was absolutely very little change, nothing happening. When we put the different energy waters into this system, each one of those waters gave us a different, unique pattern of a signal. And so the thought was that the information is not the same that's in them, right? So a water that's supposed to increase brain health was very different than a water that was targeted for immune health. So one of the first experiments we did with these infoceuticals and some of this immunity water that we had in the laboratory was to see if somehow there was information that this frequency then trapped into this water through this imprinting mechanism of immunity. And so we exposed the cells to this immune water. We did multiple dilutions, I think out to like a 1,000,000-fold dilution of the concentrated material. And then we let that information sort of vibe into the cells for about an hour. In an hour, then we challenged them with this pseudovirus that represents what SARS-CoV-2 infectivity would do. And then 24 hours later, we looked at red signal. And to our surprise and amazement, what we saw is that the cells that were treated with the immune water had less red signal, meaning that there was less SARS-CoV-2 pseudovirus that got into the cell. So there was some information that was captured through frequency imprinting into that infoceutical that was the immune water that prevented the virus from entering these lung cells in a dish. The other interesting thing we found is that this effect was non-dilutable, right? And so up to 1,000,000-fold dilution, the effect was still the same as if you added the straight amount of infoceutical into that water as well. When you work with a cell, it's the smallest unit of a living organism, right? It is life in a dish. And so this creates a very nice model to study new phenomenon and new kinds of drugs and therapeutics in. And so this is the model we adopted when we were testing these waters. We can do all kinds of amazing studies in cells. Cells have a membrane that you can study in excruciating detail, which is the focus of my laboratory. Cells have internal compartments that create everything a living system needs to thrive and grow and move and do all kinds of things. One of the primary systems that controls this is mitochondria. So mitochondria are super old bacteria that were ultimately incorporated into living cells. The advantage that they provide a cell is that they create a lot of energy. So you can create ATP which is the currency that living systems use to move, grow, benefit, think, create complexity. And so the thought was, with this energy driver, one of the first things that we noticed that we saw some major changes in is, could this energy driver and cell driver impact the behavior of the cell's energetic system, the mitochondria? We have a lot of fancy systems in the laboratory that can measure mitochondrial function in really, really fine detail. And so we added this cell driver to the cells, and we tested a bunch of different cell types: muscle cells, skin cells, cancer cells as well. And what we noticed is that when you added this cell driver to that cell, mitochondrial energetics went through the roof. So the mitochondria were behaving in a manner that we didn't expect. So the energetic potential of that cell increased. So mitochondria, in addition to making energy, are also the potential death of a cell, right? So when mitochondria become dysfunctional, instead of using oxygen to make ATP as the electrons move down this electron transport chain and oxygen gets utilized, they now start using oxygen to make free radicals. So reactive oxygen species get generated. These reactive oxygen species then damage mitochondrial function. They damage DNA within the cell. They damage proteins. They damage lipids. Everything then starts to fall apart. And this is a hypothesis that's been proposed in a lot of different areas. A lot of people think that aging is a mitochondrial disease, right? There are reactive oxygen species that create this aging phenotype. What we noticed with treatment with these various waters is that you could suppress the reactive oxygen species generation in mitochondria. And so when we look at the effect of water in cell biology, one of the things you have to think about is the primary source of how this information gets transmitted to the rest of the cell is the membrane. The membrane is a complex mixture of lipids. There's lots of proteins in there. There's ion channels. There's effector molecules. There's receptors. All kinds of things that biology needs are at the cell surface. There's lots of evidence to suggest that as information gets taken into this cell surface, it then gets transmitted into the cell, into the nucleus to change the gene program and then remake the cell in terms of how it behaves. And then there's lots of evidence to suggest that the membrane then targets organelles like mitochondria and other things. This has been the focus of my laboratory. So we're very interested in how membrane behavior and how membrane oscillations are impacted by outside forces. So the obvious thought was, does this water impact the membrane structure and behavior in unique ways? As we did this interesting experiment initially, we added water, and then we can label lipids in the membrane with these particular spin probes. So we went back to this EPR, this electron paramagnetic spin resonance. We labeled the cell membranes with this lipid probe, and then we treated the cells with water, and then we probed to see how these lipids are moving. And what we noticed is that the structure of the membrane changes. So the information that this water contains now changes the behavior and organization of the lipids, the proteins, the ion channels, everything else that's at that cell surface. So we think that this is how information is being communicated from the water hitting the cell on the outer membrane and then changing the behavior internally. Growing evidence suggests that as the cell membrane has unique compartments, and one of these compartments is a cholesterol-rich compartment that's more rigid, less fluid. And lots of evidence suggests that as you make more of these rigid compartments full of cholesterol, you can actually change the survivability of the cell. You adapt the cell to more stressful environments. What we noticed is when we added this water to the cell, we created more of these rigid environments within the cell membrane, suggesting that this is one mechanism by which the water is adapting cells to survivability. It's difficult to extrapolate cell-based data to human, but what this initial finding would suggest is that when cells are treated with these various waters, you can modulate the energetics of that cell. If this is true and extrapolates to the whole person, this would suggest that energy drives everything, right? Energy is how we endure, how we grow, how we survive, how we adapt. And so the contention is, if we can scale this up, this is where we're headed, that we're really manipulating and translating and transmuting how energy is handled in a living person. To a skeptic, I would say look at the data, right? When most people talk about water changing life and behavior, people's eyes roll and they think of snake oil and other things. The real data and the data that we're generating suggests that there's something unique about this water, that information can be captured, can be transferred, and can ultimately augment the quality of a living system. And that's what we're looking at, is look at the real data, look at the hard data. Again, you can look at it with a skeptical eye, but see how well it's controlled, see how well it's done, and then ultimately look at what the effects are.